Frederick Sanger was one of the few scientists who actually won 2 Nobel Prizes, and back in 1980, together with some other people, he won the Nobel Prize for figuring out a method on how to determine the base sequence of DNA. So this is therefore also called as the Sanger sequencing method, which I would like to explain to you. And you need 2 things in order to determine the base sequence. You need PCR, polymerase chain reaction, which copies DNA, and you need gel electrophoresis. And I'm now going to tell you how we need to both of these methods to determine the base sequence. So what is the problem? The problem is that I have a little tube over here, okay, with in here with some DNA, okay, and I give you this tube and I ask you, please tell me what is the base sequence of this DNA. And of course, by just looking at it, you don't know the base sequence. So you need a method, and Frederick Sanger, he figured this method out, and that's what I'm going to explain to you now. So what I have over here, this is the unknown DNA sequence, okay? I mean, we know it now because I told you, but we now assume that you don't know this, so that's the unknown DNA. Unknown DNA sequence, and what are we doing? What I do is I have this unknown DNA sequence. I separate this and have it in 4 different little tubes, little containers, okay? I separate them, 1 for each base. So these are supposed to be the little plastic tubes, which each of them contain my DNA. Okay, here at the bottom, the same one, right? The unknown DNA. And then what I do is the following. I do a PCR with each one of them. I copy the DNA. I copy the original DNA using PCR. But this is not, but what I'm doing here is I'm not copying the total DNA, but the PCR is modified in such a way that the first one will always stop copying at an A. So when I do a PCR, then I will do the following. It will simply copy this DNA, and it will copy it and will make a C, G, T, T, and it will A, and it will stop here. Okay, so the last one is an A. Okay, and the reason why it stops here is because I added into my PCR reaction, I added so-called D-deoxynucleotides. And so this last one here, this is a D-deoxynucleotide, and what does this mean? This means that it will stop here because I'm missing an OH group, okay? And what this means is that the enzyme is not able to continue to copy the DNA. And you know what I'm doing right now? I'm actually checking whether I spelled D-deoxy correctly, because it might be also called a di-deoxy because it's missing 2. It is missing 2 OH groups. Di-deoxy, of course, not D-deoxy. My intuition here proved me correct. It's di-deoxy, of course. Okay, because di refers to it. It is missing 2 hydrogen, not 2 hydrogen, 2 OH groups. Okay, di-deoxy. Okay. Sometimes intuition is a reliable way of knowing. I'm a TOK teacher as well, of course. So this one over here, what about T? I copy the DNA, C, G, and it will stop here at T. Okay, but there's a second T over here. Now I'm running out of space. C, G, T, and this T over here as well. Okay, and it will also stop. We have a T over here, here, and here as well. So we have. I'm not going to copy everything here now. Okay. We'll copy everything, and until we have a T over here at the end, it will copy everything until we have a T here at the end, yeah, and everything until we have a T here at the end, okay? Yeah? And in here we have the normal nucleotides, right? The normal deoxynucleotides, but sometimes the PCR builds in these. Actually, these are defective nucleotides which cause the enzyme to stop copying when it's built in. Okay, and the same was for G, and the same one for C. So okay, so for the G, we'll copy it all the way up to here, G, because we've got the G up here, right? And that's it. And for C, we have a C over here, okay? then we copy all the way over here until we have a C over here, and we have a C over here. Okay, so that is the result of the PCR of our PCR-C reaction. Okay, so we have we're adding different dideoxynucleotides into the tubes. Okay, we're doing the PCR reaction, and the PCR will copy it, and they will produce different lengths of DNA. Okay, each one of them has this dideoxynucleotide at the end. And so what we're doing right now is the following. So that was step one, it was the PCR, so we finished that. And now we're going to go into gel electrophoresis. And what I'm doing right now is the following. You have to understand that in gel electrophoresis, the long DNA fragments will not move as far as the short ones. So the short ones, they move very far in the gel. So what I do is I take the DNA over here in this tube, okay, and I put it into this slot here at the top. Okay, so there's a little slot, a little hole, and I put the mixture in here. And what will happen is that because this has an intermediate length, okay, the DNA, when I apply over here a negative voltage and down here a positive voltage, the DNA will move from the negative voltage to the positive. It's a wire, okay, there's a wire over here, okay, with a negative voltage, and the DNA is also negative, so negatives and negatives kind of repel each other. And what happens is that because this one has an intermediate length, this DNA, it will move all the way up to here. Okay, and you see that this one is black over here, right? Yeah, that's the DNA. Yeah. I will do the same thing for the T. I will take this T mixture over here, I put it in here into the T, and what I see is over here I have a very short one, that's this one over here, and that is this must be this one over here, right? Okay, then I have a second one which is a slightly longer, that is not moving as far, that's this one over here. Then I have yet another longer one over here, this one is this one over here. I'm running out of space to make my little arrows. And then I have this one over here, which is this one, and the longest one, that's this one over here. Okay, and I'm doing the same with G. Okay, so I'm taking this one here, I put it in here into G, and I see that's the one over here that I have, right? And over here for C, I have a very, very short one. Okay, that's this one, ends up being this one over here. Then I have this one over here, this one here, and I have a very long one. That's this one over here, and you see that it really doesn't go very far. Yeah. And the A, of course, they're over here, and the A, there is nothing over here. Just wanted to indicate it that that's the place where I put my DNA. So that is the result that I get, okay? And that's my gel electrophoresis gel. Yeah? And when I get something like this, all I have to do is I have to read it now to determine the base sequence. So I'm going to erase this here again to make it easy, and I start at the top. And what I do is the following, I simply will start here and I will go at the top, and the first thing is a C, G, T, T, Z, G, T, T, A, T, A, T. The next one is C, that's correct, yeah. T, 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 C. And it goes on, okay? So what I do is I'm simply reading off the whole thing, in the correct sequence, and then I have my DNA sequence, okay? And I didn't know, of course, at the beginning when I did my PCR, the lengths of the DNA fragments, but I now know it. Now at the end of the gel electrophoresis, I know the different lengths, and because I know that this one over here must end in a C at this length. Here, this one must end in C, this one must end in a C. This one over here must end in an A, and so on, okay? Because the last one is always this dideoxynucleotide, which is not able to continue making DNA. Well, if this one is built in, then the polymerase is not able to continue to make DNA. Let's put it correctly like that, okay? And that is the reason why back in 1980, Frederick Sanger, he got the Nobel Prize for determining this. And this system is still used, of course, nowadays. What you have is you have a machine that does that. You don't have to read it off yourself by hand anymore, but the machine, you drop it into the machine, the original DNA, and out comes the DNA sequence on the computer monitor, right? So this is really a very standard and very automated procedure nowadays to determine base sequences. Everything's basically done now, automated, to also to minimize the mistakes, that so that humans cannot make any mistakes. Very important, not only for research but also for forensics sometimes. Yeah, and this is how they have determined the base sequence of the human genome, and this is how you can determine also what the different parts of the DNA in a certain organism are doing, because if you know the base sequence, then you can also determine the proteins that this base sequence codes for. And there's a whole huge area of research called bioinformatics that deals with that. And bioinformatics is the use of computers for biology. Okay, because you can do all sorts of fancy statistical analysis using the DNA sequence. You can search for genes. You can do, I don't know, sequence comparison. You can compare how similar is organism A to organism B. Lots of things. Very, very useful. A very useful method. Okay. And the side issue, because I already said it in 1980, Frederick Sanger got the Nobel Prize for this. Kerry Mullis also got the Nobel Prize for inventing the PCR method. Okay, so the PCR is there to copy the DNA, and that is also a very, very central method in molecular biology. So for that, there was also a Nobel Prize awarded. So that is basically it. And if you think this whole thing is kind of complex, then I would say the minimum, at least you should know how to read off a gel like this, okay, to determine the base sequence. I mean, that is really the bare minimum, okay? And also know that you need PCR and gel electrophoresis for the Sanger sequencing, and you also need these special dideoxynucleotides which stop the copying of the DNA once they're built in, okay? So just to clarify, all the other nucleotides before, they are normal nucleotides, even here in the T, okay? These are the T over here, which is before the final one. That's a normal nucleotide. But if the PCR starts to use a dideoxynucleotide, then the polymerase in the PCR is not able to continue to make it, right? And then it stops here, okay? And that's basically it, okay? So a very useful technique that is used in molecular biology very much, but nowadays I think nobody actually does this by hand anymore. You have machines that do that for you. All the best, all the best.